So there's a sort of large interest in the oceanographic community to understand the biology of these oxygen minimum zones. And so multiple groups have characterized the bacterial community composition associated with oxygen minimum zones. This is just a big figure from a very recent review article in Nature Reviews Microbiology. And the point of this is if you look at different bacterial lineages along here, you see that there's a striking change between oxic waters, so surface waters, and oxygen deficient waters. So there are sort of these oxygen deficient bacterial specialists that thrive under these conditions. And one of these oxygen minimum specialists is this clade of bacteria called the GSO clade. And this is a phylogenetic tree. Looking at the GSO clade, the sequences in here are just color-coded based on different oxygen minimum zones. And the point is, these guys are globally distributed and abundant in oxygen minimum zones worldwide. So they're an interesting target group to do metagenomics on to try to figure out what they're doing. I'll just say that this GSO clade, which is the whole group here, is actually comprised of two subclades. One is called SUP5. I'm sorry about the names, but I don't name these things. We don't have cultures, right? So we have all these sort of abstract clade names. And the other one is this Arctic 96BD19 clade here as well. Okay? And I'm going to talk about the ecological differences between these, so that's why I'm pointing it out. So a few years ago, working in one of these oxygen minimum zones, we were able to use metagenomics to reconstruct the metabolism of one of these organisms, particularly the SUP5 group. And so we were working in Saanich Inlet, which is an anoxic basin off the coast of British Columbia at the time. And what we were able to do is, from whole genome shotgun data from a mixed community, we could actually assemble that data, and then, so using bioinformatic approaches, actually bin the contigs that originate from the SUP5 population. And so we reconstruct this genome basically from the environmental sample. And what's nice about this is now we actually have all of the genes, 1,300 of them or so, in the same bag, right? So we can reconstruct the metabolism of this organism in the environment. And so this is just a schematic looking at the metabolic reconstruction. And some of the highlights here, I don't want to go into all the details, but we could demonstrate that this organism had the ability to fix CO2 via Kelvin cycle. It has an energy metabolism that is based on the reduction of sulfur compounds. So hydrogen sulfide, for example, accumulates in these oxygen minimum zones. This organism actually has the ability to detoxify this hydrogen sulfide. So it's interesting from that perspective. And also, it doesn't grow aerobically, but instead it uses nitrate as a terminal electron acceptor. And also, its genome gave us evidence that it might actually produce nitrous oxide through its metabolism. And so nitrous oxide is a potent greenhouse gas that is produced in these oxygen minimum zones, and this might be an organism that's actually responsible for that. Okay? So that's just the kind of the idea with how we can use metagenomics to describe the metabolic potential of these organisms. Okay, post-metagenomics. So the problem with the genome is that it only reveals the genetic content and the metabolic potential of an organism, right? It doesn't tell you anything about what that organism is actually doing in the environment or how the metabolism of that organism is varying across some sort of environmental gradient, okay? So we need ways of actually detecting activity under in situ conditions, okay? And so there's gene expression and protein expression approaches, so like transcriptomics and proteomics. And the way I like to think about this is sort of metagenomes tell us about community genotype, and then as we move through the central dogma of biology to metatranscriptomics, metaproteomics, we're getting more information on the actual phenotype of these organisms, right? Because we're looking not just at the genes, but the actual proteins that are expressed under some set environmental conditions. And so in the last 2 years or so, I guess, we've started using metaproteomics to look at gene expression patterns of microbial communities in the oceans. And our most recent study was actually done in Bedford Basin. And here we're using metaproteomics to look at changes in metabolism in microcommunities over time. And so for this study, we picked sort of 4 distinct samples to work with. We took samples in the winter, surface and deep. And in the winter, Bedford Basin is very interesting because if you look at the oxygen profile here, it's actually hypoxic in the deep water. Okay? So it's a target system for us to look at how some of these oxygen-deficient microbes might be responding to different environmental conditions. And then we took samples in the spring. Okay? And here you can look at fluorescence, which is telling you about the chlorophyll. We took these samples that just occurred after this phytoplankton boom. So we can look at how microbial gene expression changes over the course of a phytoplankton bloom, and how also it's regulated in surface waters versus deep. Okay, so I put this slide in, this very busy slide, just to show the sort of workflow for metaproteomics. And the take-home message is it's the same as doing a metagenome, but you're just extracting protein first, right? So we start with a sample, we extract proteins, and then we use tandem mass spectrometry to actually generate the data on those proteins, okay? So that's the first step and the second step. The real trick here in metaproteomics is the third step. And the reason for that is because to identify proteins that are expressed, we have to take the mass spec data and search it against protein sequence databases. You can only identify proteins that actually exist within your protein sequence database. And so for us, right, dealing with these complex environmental communities that are changing over time, we basically do the best we can. So our protein sequence databases have reference genomes from all isolates that are in the oceans. We have metagenomic data from the same environment, and then we also have reference genomes from whole genome amplified single cells. So I haven't talked about that, but sort of one really important technology that exists now is sorting single cells, amplifying those genomes, and then sequencing those genomes of single cells. And we could use those as references for identifying proteins that are expressed in the water column. Okay. So if we look at the 4 samples, the actual data that comes off of our mass spectrometer is about 592,000 spectra. Okay? So that's the raw data. When we annotate that against our databases, we only identify 48,000 peptides. Okay? So only about 10% of the data we're actually able to identify what proteins it comes from. So this is a major problem with proteomics right now, and it's one that we're dealing with, but you have to keep in mind that we're only looking at a small fraction of the data that we actually generate. Nonetheless, that 10% actually tells us a lot about what's actually happening in the community. So here we're just looking at the taxonomic variation of the 4 different proteomes. And what you can see is that there's these different phylogenetic groups are expressing proteins in different samples, right? So we see that there's this phylogenetic split between proteins that are expressed. But more interesting is to actually look at the metabolism of these proteins. So what are these proteins doing? And so one thing that was very striking is that we see from winter to spring, in surface and deep waters, we see a massive increase in membrane transport proteins. And this is pretty cool because these transporters are all specific for different compounds. And what we're seeing here is basically a readout of what compounds these microorganisms are actually using in these different samples. So for example, in the deep water, underlying the spring phytoplankton bloom, we see a high expression of amino acid transporters. So this tells you that amino acids are important to the metabolism of these organisms under these conditions. And then there's some other sort of interesting stuff like glycine, betaine, and taurine. These are compatible osmolytes that are produced by phytoplankton. They're also, you can imagine, a rich source of nitrogen, for example, for microbes, and we can identify the transporters for these compounds. So it's giving us a readout of what actually these organisms are eating in the water column. So we also found some kind of unexpected stuff. So we found genes for methanol oxidation in the coastal ocean. And methanol oxidation is well known to occur in bacterial populations, but it wasn't known to actually be occurring in the oceans. And so during the spring phytoplankton bloom, we actually see a high increase in this methanol dehydrogenase. And these are proteins from a group of organisms called OMZS that are known to be methanol oxidizers. So we identify these in the oceans. And the immediate question that comes up is, so what is the actual source of methanol in the oceans? And right now it's actually sort of unclear. There's potentially phytoplankton sources for methanol. There's also methanol that's actually produced by terrestrial plants, and it actually might be deposited into the oceans as well. And so we're sort of following up on how methanol is produced, how methanol is consumed in the oceans now, just because we see these proteins. This is just looking at a time series of this OMZS group, and you can see that in Bedford Basin over the full year, they're there all around. Okay? But of course, they're more active in the spring. Okay. So I'm going to skip this guy because we didn't talk about it, but we find genes for archaeal ammonia oxidation and autotrophy as well. But I will come back to this one here. So I told you about the GSO clade. This was the clade that the SUP5 group is an organism that fell within that used sulfur compounds, fixed carbon dioxide. In Bedford Basin, this red guy here, this is the hypoxic deep water, and what we can see is we actually find proteins that are actually expressed from these organisms. And so this is interesting to us because what we want to know now is what are the functions of the protein, and do they tell us anything about what these groups of organisms are doing under these hypoxic conditions in Bedford Basin. So if we look at the SUP5 proteins first, so remember there's two different populations here. One of the interesting questions is what is the difference between SUP5 and Arctic? Is there ecologically distinct populations or the same? So first what we did is we looked at the SUP5 proteins that were expressed. And the most striking observation was that we found strong evidence for sulfur oxidation actually occurring in Bedford Basin under these hypoxic conditions. So it's not important that you understand the actual pathway here, but the green enzymes are enzymes that we did detect, and the red enzymes are those that we did not detect. And so what we can say based on this pattern is that SUP5 under the conditions in Bedford Basin are able to use this compound as an energy source, this is thiosulfate, but are unlikely to use hydrogen sulfide. And this makes sense because in Bedford Basin, actually, we don't expect hydrogen sulfide to be accumulating, but this organism is sort of fitting the pattern that we expect, and we see these genes turn on. Interestingly, we don't find any of the genes for CO2 fixation, so it's unknown at this point how this organism is actually acquiring its carbon in the system. And we didn't find any of the genes for nitrate reduction pathways being turned on as well. So we can really only say that this is a population that is performing sulfur oxidation, but we don't know much more about its carbon and nitrogen pathways. So the next question of interest is, okay, so that's fine, these guys are doing sulfur oxidation, but what are these guys doing? We identified proteins there as well. And you just have to back up a little bit and look at what other people have reported in the literature on the metabolic capacity of this group. So this is what people know about sequencing single-cell genomes or sequencing metagenomic data from this organism. They have CO2 fixation pathways and sulfur oxidation pathways, so similar to SUP5 in the metabolic potential. But they also have many high-affinity ABC transports for organic compounds, meaning that they can use organic carbon compounds to generate biomass. And so we looked through our proteins to sort of figure out what these organisms are really doing under the conditions in Bedford Basin. And for this group, we identified all sorts of transporters for amino acids, dipeptides, sugars, and taurines. So it looks like this organism is actively importing organic compounds into its cell. And we did not detect any of the proteins for sulfur oxidation or CO2 fixation. Okay? So these genome papers sort of report on the ability for these organisms to do these processes. But it's really important, I think, for these proteomic data sets to sort of come in and try to figure out under what conditions are these pathways actually turned on and off. And of course, that has ramifications when we're thinking about carbon cycling, sulfur cycling, and nitrogen cycling within the environment. Okay, so just to wrap up, so using the proteomics, we were able to show that the metabolic pathways that are associated with spring phytoplankton blooms involve ABC type transporters. So we see this upregulation of proteins for the consumption of carbon compounds that are probably associated with the phytoplankton bloom. We found these novel C1 compound pathways, so like methanol, that are detected in the water column. And during the winter, under these hypoxic conditions, we find a dominance of chemolithotrophic metabolism. Okay, so these are organisms that are not using organic carbon in the system. So the chemolithotrophic sulfur oxidation by SUP5 is one example, and I sort of skipped over the Archaea, which are another very interesting group of organisms in the oceans that have the ability to use ammonia as an energy source and use that energy source to fix carbon. And we see them in the system as well. Okay. So thank you. I'd like to thank Hiba El-Swayes is a wonderful student who just finished her master's in my lab and is now going on to do a PhD. She worked on all the time series work in Bedford Basin. And Anna Georges was an exceptionally talented undergraduate who actually worked in my lab and did all of that metaproteomic work on her own. She developed the methods, she developed the bioinformatics as well. And is now going to the University of Toronto to do a PhD. And our important collaborators on this project are, of course, the people that are running the Bedford Basin time series, most importantly, William Lee, who's at the Bedford Institute of Oceanography. And we're also collaborating with people at Dalhousie University, developing novel statistic approaches to characterize bacterial plankton communities in the oceans. Thank you.